I looked into, that was perfect transition to the next question, about microRNA. I mean, from what I heard, one specific microRNA can impact hundreds of different messenger RNA. So it's kind of a massive cascade. And if that's also, I think, why, a lot of drugs were very hard to target the microRNAs, because they're just such a big impact, and whatever pathway could be impacted, leading usually, I guess, to pretty high toxicity. But so how, and I also, at least from what I've seen, that you guys were basically one of the first microRNA targeting drug, at least in cardio, where it really seems to work better versus all the trials where, just what I said, that just too high toxicity. So, what specifically did you figure out, basically, to make it work versus other microRNA targeting?
I think in first instance there was a different approach to identify the right microRNA. So in Thomas' labs, he did over many years structured wet lab experiments, robot-based screening of hundreds, thousands of microRNAs, and finally found that one which he brought from the cell culture dish into patients now over the course of a very contingent pharma GXP compliant process. But the difference was we are sometimes asked whether there are other groups that have also been doing research in 132. Yes, that has been done, but finally the whole IP that is around that was filed in 2012 with the first patent family by Thomas to the medical school Hanover, and Cardior has licensed that exclusively. And we have built a large protection around this asset. But what makes it unique, I think, is also the chemistry. We have been working with different chemistries until we found that specific locked nucleic acid chemistry. And that has a specific spiking pattern that stands for low toxicity. So before we brought it into animals, we have been ensuring that the chemistry is a real optimal one. And there were effective knockdown experiments. The target is 100% conserved, really from fish to worm to mice to pigs to human. That is a guarantee for the translation of the different results you get. This is not the case in every microRNA, but with 132, that was successful.
Okay.
And also this...
You're down-regulating it, right?
We are down-regulating it. So we are blocking this microRNA.
Okay. Yeah.
So you have, once the cardiac muscle gets into stress, being it a myocardial infarction, microRNA 132 levels are increased. Yeah. And what we are doing is we target the microRNA by having an antisense oligonucleotide. Yeah. That is a 16-mer oligonucleotide based on a locked nucleic acid.
16 base.
16 base. Yeah. With a phosphorothioate backbone. Yeah. And it's a very stable chemistry. So and this goes directly into cardiomyocytes, binds to the 132 microRNA, blocks it for 4 weeks or the like.
Okay.
It does not destroy the target. It's not like an siRNA which destroys the target. So there is a difference. Yeah. And this binding, which is repeated in our current trial for 3 times. In the more chronic setting, it will be repeated for 6 times. So every month, the patient will receive an injection, and that should be sufficient, which has been studied and showcased in these large pig trials. So we had 2 different animal models, one with the subacute setting and one with a chronic setting.
Okay.
Yeah.
Okay. And that I'm curious, just do you know exactly? You say blocking, but I guess you can also quantify or measure the amount of mRNA left.
Yeah.
You're blocking, or like
we are blocking
down again, or blocking.
We are reducing the target with more than 50%, 55%. Okay. This is then also the PD active scenario. If you use a dose that is able to reduce your target more than 55%, then you are effective. Yeah. And in our studies, we were also able to show a knockdown of 80 to 90%.
Okay. Yeah. But I guess you still want,
but if you want to dive into more detail, you need to ask your CSO.
Oh, good. No, I mean, I'm curious also. I mean, as from the beginning of the discussion on the microRNA, I mean, as it impacts so much, I guess there are also some other function, even MI132 that has some functioning, that it might be good to not knock down 100%.
Yeah.
Just
that's true.
That was my thought. But yeah, I like that because so if I summarize really the target, but also the chemistry and how you attack how you target the target. Because I remember microRNA, Abivax was on the show, but they target with some small molecules, also target microRNAs, but with small molecules seems to work also pretty well. But I mean, they're running a phase 3 at least, but we'll see how this goes.